All right, so we're here now with our second section on Microevolution, which is chapter 20 in the Raven book. And you may have noticed that that first section took quite a bit of time for the number of slides we got through. It was about 20 minutes for only about 8 slides. And that was because that first section really sets up what's going to happen in the rest of this chapter. So it's really good to have a nice, firm understanding of that first section before we move on to this next little bit. And throughout the rest of the chapter, really what we're going to be doing is going through and talking about some more details about mutation and gene flow and genetic drift and non-random mating and natural selection and how they participate in contributing to the whole process of microevolution. So when we look at the mathematics of this, now this is an equation. There is not going to be any calculations that you actually have to do with this. So right off the bat, if you're not a math person, don't panic. We're not actually calculating anything, but looking at this equation and how it sort of pertains to the process of evolution and gene frequencies, it's a really useful way to sort of look at things from a different perspective and see how this process actually works. So the Hardy-Weinberg equilibrium equation is, for whatever reason, by convention, going to use the alleles P and Q. Kind of by convention, historically, and by tradition, they always use P and Q for the two alleles that are going to be tracked using this whole process of mathematical calculation. So, in the example that we're going to use a very simple example that looks at coat colors in plain old house cats, right? So first off, this is by no means a realistic example of what actually happens here, but if you sort of just sort of use your imagination here and just kind of let the whole taking everything really, really literally kind of go for a few minutes, it actually is helpful to help understand how this whole process works. So in this population of cats, we have two alleles. We have P and we have Q. Now the P allele is the dominant allele and the Q allele is the recessive allele. P is essentially a big B for black coat color. All right. Now, what that means then is for any black cat that we see in this population, it could either be homozygous or it could be heterozygous. And in either case, regardless of what the genotype is, the phenotype of those cats is going to be black. Meanwhile, any cats in the population that are white, because the white allele is the recessive allele, we know that those white cats have to have the genotype of homozygous recessive. And so if we take all three of those genotypes and add them up, if we take all the homozygous dominants and the heterozygotes and the homozygous recessives and add up those percentages or add up those frequencies in the population, they should add up to 100%, or if you're using decimals, they should add up to 1. All right, because every cat in this hypothetical population has to be either black or white. That's the only options we have in this population. Now, in terms of Hardy-Weinberg, the homozygous dominants are represented by p squared, the heterozygotes are represented by 2pq, and the homozygous recessives are represented by q squared. All right, so this is actually the equation we're going to be working with as we go through this example. All right, so in this hypothetical population of cats, we are going to pretend that we go out and capture them, and we do genetic analysis, and we find out the genotypes for all of these cats in this entire population in terms of their coat color. And so what we find out is that 36% of the cats have a homozygous dominant genotype, 48% of the cats have a heterozygous genotype, and only 16% of the cats have a homozygous recessive genotype, and therefore they have the white phenotype. So if we take this 36% plus half of this 48%, 36% plus 24% means that in the entire population of bees in this population, 60% of them are the dominant big B allele. And if we look at the little b allele, 16% plus half of the 48% gives us 40%, or if we just do 1 minus 0.6, that also gives us 0.4. It's maybe an easier way to do it. 40% of the bees in this population are going to be of the recessive type. Now, what you might be wondering is, well, why do we do the half of this value here? Well, if we look at the genotype here, these cats have both a big B and a little b. So for this 48%, 24% of the genotype goes the big B way, and 24% goes the little b way and goes over here. So 24% plus 36%, 24% plus 36% gives you 60, and then 24% plus 16% gives you 40% over here. All right, so that is our generation 1 of our house cats that we're starting off with our analysis. Now, based on that, we know that 60% of the sperm cells in the population are going to carry a big B, or the P allele, and 40% are going to carry the little b allele, because those are our percentages here, right? So big B was 60%, little b was 40%. So 60% here, 40% here. Same thing with the egg cells, 60% here, 40% there. And so what we do is we then figure out the frequencies or the probabilities of each one of these sperm cells fertilizing any one of these egg cells up here. So 0.6 plus 0.6 leads us to suspect or predict that in the next generation of house cats, 36% of them, which is 0.6 times 0.6 is 36% should be homozygous dominant. If we do 60% times 40%, that gives us 0.24 or 24%. Same thing here, 0.4 times 0.6 is again 24%. So these two genotypes are effectively the same thing. They're both heterozygous. So we can actually add these two together, and that's going to give us 48%. And then if we do 0.4 times 0.4, that gives us 16% here. So that gives us a frequency or probability of having 16% of the bees in the next population for coat color in cats of being homozygous recessive. All right, now what we want to think about, okay, in generation 2, how do our frequencies compare to generation 1? What we're saying in generation 2 is that 36% are homozygous dominant, 48% are heterozygous, and 16% are homozygous recessive. Well, let's go back and look at generation 1. Generation 1 was 36%, 48%, 16%. Generation 2, 36%, 48%, and 16% down here. So has the population changed in the gene frequencies? No, it hasn't. They're exactly the same as what they were before. 36, 48, 16. 36, 48, 16. They haven't changed. So therefore, we know that evolution has not occurred in this population. Now, what would be the case if the frequencies had changed? If the homozygous dominants or the heterozygotes or the homozygous recessives, if those frequencies had changed at all, then we would say that, yeah, our population has evolved. Well, this is where a population geneticist or evolutionary biologist or both would be interested in figuring out, okay, well, what actually caused the change in the frequencies? Was it some benefit of being or having black coat color? Is there some benefit of having white coat color? Is there some benefit of being a heterozygote? If these frequencies change from generation to generation, if evolution occurs from generation to generation, if we go back to our Hardy-Weinberg assumptions of no mutation, no gene flow, no genetic drift, random mating, and no natural selection, those are the 5 agents of evolutionary change. So that if we see any change from generation 1 to 2 to 10 to 100, whatever, if there's any change in frequencies at all, we now have 5 suspects that we can then sort of pin down the cause of this change on. And so maybe there was natural selection, or maybe there was some non-random mating, or maybe there was a mutation, and all of a sudden we had brown cats showing up, or any of those kinds of things, drift, or maybe there was some migration of cats into or out of the population. One of those 5 things, or more than one perhaps, might have caused those frequencies to change. So really what the Hardy-Weinberg equilibrium is a control model that illustrates what happens if a population is not changing. If a population is not changing, that's the same thing as saying that it's not evolving, which is the same thing as saying that it is in equilibrium. Now again, most populations are not going to meet those 5 assumptions. No mutations, truly random mating, no drift, no gene flow, and then no natural selection. Most populations are going to violate at least one, if not more than one of those assumptions. So what that really means is that populations are evolving all the time. Most populations are not standing still. Most populations are not going to maintain the same gene frequencies generation after generation after generation. Most real-life populations are going to change in some way as a result of one or more of those 5 different agents of evolutionary change. All right, so that's why this model is important, is it allows us to see what the population would look like if it wasn't changing, and if it is changing, which one or perhaps more than one of those 5 assumptions might actually be causing change to occur. Now, mutation by itself is the least likely to produce any sort of evolutionary change because we have talked before a lot about how uncommon mutations are, right? They're very, very infrequent. Most of the time mutations get fixed, even when they do occur. And most of those mutations that don't get fixed end up not really having any sort of effect on the phenotype, and so therefore they don't really matter as far as a natural selection perspective. So in the grand scheme of things, mutations don't really add up to very much in the whole evolutionary process. But mutations do sometimes add new alleles that natural selection can either favor or select against, and therefore produce some more powerful evolutionary change. All right? So mutations are sort of like seen as the fuel to evolution. They add new alleles that natural selection can pick and choose from. Gene flow, like we talked about before, occurs when you have migration of individuals into a population or out of a population. And so when those alleles are moving into a population, if they're immigrating coming in or emigrating going out, so unfortunately those two terms sound very familiar or very similar. Immigration means coming in, emigration means leaving. Either way, if either of those two processes is occurring, then the alleles carried by those organisms might change the frequencies of that particular population. So if you have, for example, if for whatever reason mass exodus of black cats leave the population, that's going to change the frequencies in the population. Or the same thing if it's white cats leaving the population, right? Now, in this example, it doesn't really make a lot of sense that that would actually occur, but you see what I'm getting at, right? If there's migration, then that can kind of throw off the allele frequencies in a population. If you're dealing with species that reproduce over a wide area, like plants that are pollinated or fungi that produce spores, again, very widespread migration, not of individuals so much, but of gametes in the form of pollen or spores that can cause some pretty large-scale gene flow as well. Now, non-random mating actually comes in two forms. Non-random mating can be in the form of either assortative mating, which is where individuals choose to mate with those individuals who are somehow similar to themselves, right? So in our example of the cats, assortative mating would be if black cats preferentially mated with black cats and white cats preferentially mated with white cats, that would be an example of assortative mating. And the interesting thing about that particular type of non-random mating is that it can potentially divide a population to a point where, if other conditions are met, those two different types of cats, or two different types of phenotypes, if we're not looking at cats, could actually cause the population to split in two. If the black cats only mate with black cats for a really long period of time, and the white cats only mate with white cats for a really long period of time, any other differences between black and white cats, aside from coat color, might eventually cause black cats and white cats to diverge into two separate species. Now again, in a literal sense, for this example, that's probably not going to happen, but it is illustrative. It's a very simple example, and it sort of shows us what kind of power assortative mating has when it comes to creating and maintaining and really accentuating differences within a population when you have that assortative mating going on. Now with disassortative mating, this is the opposite, okay? So disassortative mating is where individuals choose to mate with individuals who do not look like themselves or do not resemble themselves in some way. So if black cats preferentially mate with white cats and white cats preferentially mate with black cats, then that would be a disassortative mating type situation. Now, disassortative mating has the opposite effect on the heterogeneity of the population. Because if black cats and white cats are always mating together, any sort of other differences that are in those black cats or white cats are going to get mixed together, and the population becomes more genetically homogeneous. So in general, assortative mating tends to lead to more exacerbated differences in the genes or the gene pool of a population, whereas disassortative mating tends to blend everything together and create a more homogeneous population from a genetic standpoint. Now, genetic drift is one that can occur a couple of different ways. This is probably the one that's maybe the most requires the most understanding, the most explanation, and the most sort of thought on your part to understand how this works. So genetic drift is essentially what it amounts to is random changes that occur in a population that are not the result of any of the other four factors. And random chance events are going to have the biggest impact on small populations of species. All right, so let me give you an example of that. One type of genetic drift is called founder effect. Now, the term founder effect, what that means is like if you imagine somebody who founded a new town, or somebody who founded a new territory, somebody who founded a new business. It's a establishing sort of a situation taking place here. A founder effect is where you have a new subpopulation that comes out of a bigger population and colonizes some sort of new territory, like what's happening in the birds in this picture up here. So these birds have founded a new population on this little island, which is separate from the mainland. Now, the reason why I said that these populations, small ones tend to exhibit drift more frequently than big ones, let's look at the numbers here. Let's say that we've got, like literally, in a very literal sense, 5 birds on the mainland. 4 of them are red, 2 of them are yellow. Now, if you were to pick 3 birds out of the population and say, okay, you guys are going to be blown out to sea in a storm, you're lucky enough to land on this island and create this new population here, there's a very good chance you might pick just red birds if you literally only have 6 birds to choose from. you might possibly get all red birds by picking 3 out of that original 6. Just like you can get 3 heads in a row by flipping a coin. Same kind of idea, right? It's not necessarily likely, but it can happen. Now imagine what would happen if we had 6,000 birds on this island. Now, if we had 6,000 birds on this island, how likely would it be to pick, well, I guess in that case, 2,000 birds and put them out here on this island out here, right? So it's literally not as likely to happen that way because you're choosing more from the original population and moving those out to sea. So in a big population, you're less likely to get just red birds versus something that looks more like the original population over here on the mainland. All right, so that's a founder effect, and the smaller the number of founders or colonizers or establishers or whatever you want to call them, the smaller the number that gets moved out to this new population, the more likely you are to get a different gene pool here than what we had out here. All right, so that's a founder effect. Now, the other way that's kind of common to see bottleneck effect, or to have genetic drift, is by having a bottleneck effect. Let me say it again. A second way to sort of notice or exhibit or observe a genetic drift event is through a bottleneck effect. So bottleneck effect, this is kind of like the theoretical way this would work, right? So bottleneck effect usually occurs when a big population is reduced down to a relatively small number for whatever reason, and the individuals that survive and recolonize the population, their genes don't reflect the gene pool of the original parent population. So in this example up here, we've got a bottle and we've got beads in it, right? So we've got, I'm not going to count these, but let's say we've got 10% red beads and 45% yellow and 45% green. So that's our 100% of the beads. We got 10% red, 45% yellow, 45% green. 100 beads total. Now, if we randomly select 10 beads out of those 100 beads from this bottle, and we put them in this beaker to create our new population of survivors, this is the next generation that actually gets to move on and replenish the population with its offspring. In this scenario, what we've done is we've pulled out 80% green, 20% yellow, and 0% red. Now again, remember, originally we had 10% red, 45 yellow, 45 green. Now we're at 80% green, 20% yellow, 0% red. Very, very different gene pool here than when we had originally, because the ones that we pulled out of this bottle are not reflective of what was actually going on in the overall parent population. So if these individuals then recolonize and repopulate the population, the numbers are brought back up by reproduction and offspring and so forth, the next generation over here looks a whole heck of a lot different than this generation over here, right? This one was 10%, 45, 45%. This one is a whole lot of green, quite a few yellow, and red is gone. The red allele is now extinct from the population because of a chance event. It wasn't that the red was somehow not beneficial. Maybe the red was a great trait to have. Maybe it was the key to survival for this population. And just by chance, the survivors here didn't have red, the red allele is gone, and now the entire population doesn't have any red at all. Right? So that's a bottleneck effect. And this is actually a really common sort of a thing, especially in modern times when we're seeing huge reductions of species that are becoming threatened or endangered or critically endangered. The individuals that are surviving these mass die-offs of species don't contain the original gene pool that the entire population used to have. And there's actually a really good example that we'll take a look at later on in a later section of this chapter where a species was drastically reduced about 40,000 years ago. A few individuals survived, they repopulated, they recovered, they got back to almost the same numbers they were at before the die-off, but the gene pool now is very different than what it would have been 40,000 years ago because of the bottleneck effect. Elephant seals, which is what this picture is exhibiting here. Elephant seals are a sort of a unique species in appearance. Obviously, males have this sort of huge body and sort of funny looking snout. Females don't really look like that, but males are unique in their appearance. And because of their social behavior, because of their mating strategy, they actually go through their own bottlenecks, not because of a die-off, but because of how they actually breed. So if you're not familiar with elephant seals, Happy Feet 2 has actually got a fairly decent illustration of how elephant seals choose who mates with whom. It's described as a harem breeding system or situation. Harem is like a large group of females and one male that mates with all those females. So what happens in the breeding season in elephant seals is all the males gather around and they have like just the biggest, nastiest fight you'd ever see. And eventually, at the end of this fight, most of the males are either exhausted or wounded or dead in some cases. and there's one big, ugly, dominant male who's one. And that one male gets to mate with like 90% of the females. All right, so, you know, the effect of that is that that next generation of offspring is essentially everybody, well, 90% of the individuals are going to be half siblings, at least, if not more than that. So the overall diversity in the population is relatively low. Every single time there's a new generation, there is a new bottleneck that occurs because there's essentially only one male producing the offspring. All right, now there's another 10% that are fathered by other males. There's a whole story with that as well, but we're not going to talk about that. Sneaky males that look like females that occasionally get to mate with a female because the big male doesn't notice them. But that's a topic outside the realm of this class, but kind of a really cool one in an animal behavior sort of a class. But in this class, or in this case, it's an illustration of a bottleneck effect. And that leaves natural selection, right? And before we wrap up this section and move on to the third section of the chapter, third lecture in the chapter, we're going to say that natural selection is by far, 99% of the time or more, the number one agent of evolutionary change. All the others combined can't even come close to what natural selection does to produce evolutionary change. Again, the idea here, natural selection, is that some individuals leave behind more progeny or offspring than others, and those that do that have their genes more reflected in the next generation. In other words, the next generation's gene pool has changed, and therefore evolution has happened. Now, this can happen in a natural sense, like for reasons we've talked about before, all kinds of examples of natural selection, like the giraffes and so forth. It can also occur as a result of artificial selection, when you have humans that are breeding livestock or pets or plants for some kind of a trait. That's artificial selection. Humans started breeding corn thousands of years ago from a little scraggly grass plant that eventually became what we know as corn today. That was artificial selection. Individuals began breeding the corn plants that had the traits they wanted, and thousands of years later, we have something which is, really important as a food crop for a large percent of the world's population. Now, again, for natural selection to occur, we've talked about this all the way back in chapter 1, we have to have a population that is at least a little bit genetically heterogeneous, so there has to be some diversity in the population. That diversity has to be genetic in nature, so the changes that occur during your lifetime, they don't count in this context. They have to be genetic in nature, they have to be heritable in nature. And then finally, there has to be some sort of selective pressure in the environment that says that not everybody gets to survive and reproduce. And if you have those three conditions, variability, heritability, and selection, you can then have natural selection, and as a result of natural selection, you can actually have some evolution take place. All right, so what we're going to do is we're going to wrap up this section now, and we'll start up with the third section of chapter 20 in the Raven textbook here in just a second.